Hello friends and dear students, welcome to my channel Learn with Ansel. Today I am going to discuss about the RNA tumor viruses of human cancer. In the last lecture, I have discussed about the oncogenic viruses, which constitutes about the 15 to 20% of the total cancers worldwide. I discussed that most of the DNA viruses are responsible for the causing cancer in the human beings, but only two of the viruses of the RNA region are responsible for causing tumorogenic effects in human cell. One is the retroviruses, and in which two groups are there. One is the lentivirus, another is human T cell lymphotropic viruses. In lentivirus, the common virus is HIV, which causes tumors, B lymphoma and Kaposi sarcoma. And next is the human T cell lymphotropic viruses, which causes adult T cell leukemia and lymphosarcoma of B cells. Whereas the other, it belongs to the family Retroviridae. Whereas the other family of viruses which causes tumor in the human beings is the hepatitis C virus belonging to the family Flaviviridae. And in this group, hepatitis C virus causes hepatocellular carcinoma. We have already discussed that what is the oncogenic viruses. And if we define the oncogenic viruses, it is basically the virus that produce tumors in their natural host or in experimental animals, or induce malignant transformation of cells in cell culture, are known as oncogenic viruses. CE virus, which induces tumors or induces cancer in cells or host, is often no longer recognizable in the culture. Basically, we see that the effects are caused in the to study any of the viruses, we do cell culture and see what they do changes in the transformation in the cells. But usually it is very difficult to recognize in the cell culture how they transform it. But by its infectivity or its antigenicity, now here the most important is that the trace could only be found in the form of viral DNA and RNA and new antigens. Next, if we go, so it is very important to discuss about the genomic structure of the tumor viruses, to study that what is the important causes responsible for causing cancer or cancer causing agents which are present in the viruses. Here I am focusing basically on the retroviruses, which is responsible for causing the human cancer. If we classify the retrovirus, it is basically, we can again divide the retrovirus family or retroviridae family in three types. One is the oncovirinae, which contains the all tumor viruses. Then is the spumavirinae, contain viruses which are able to cause degeneration of inoculated cells, but not associated with any known diseases. And next is the lentivirinae, which can cause chronic infections with slowly progressive neurological impairment. So today we will discuss the detail about the retroviruses and also about the different type of genes or the genomic structure which are responsible. Before that, the two ways or the two mechanisms through which the virus causes is either through the activation of proto-oncogene present in the cells to the into the oncogenes, and next is the suppression of the anti-oncogene, which usually causes the suppression of oncogenic genes, which are responsible for uncontrolled multiplication of the cells. So today, we will discuss about basically the retrovirus group. So if we coming to the part, if we classify the retroviruses, it can be classified into the simple retrovirus and complex retrovirus, which is the lentivirus. And the former encodes for only three polyproteins, namely GAG, POL, and ENV, while the later encodes for six other accessory proteins, which we are going to discuss later. Now, coming to the virion structure, if we discuss about the independent virus, of the independent structure of the virion structure of the retrovirus. So it is an enveloped virus, which it is a type of enveloped viruses with a capsid that contains a dimeric RNA genome and three viral enzymes such as RT, reverse transcriptase, ion, integrase, and PR, which are packaged inside the capsid. And if we discuss about the viral genome, it is an 8 to 10 kilobase RNA forming a cap structure at the 5 prime end of the RNA and poly A tail at the 3 prime end. And it encodes for three ORFs, that is the GAG, POL, and ENV polyproteins. Coming to the genome replication, so first of all, the viral RNA genome is converted into DNA via a reverse transcription during the entry, and the DNA is inserted into chromosomes, or we can say into the host chromosome, as a provirus. Lentivirus HIV, the prototype of lentivirus, encodes other six accessory proteins in addition to GAG, POL, and ENV, that is the TAT, REV, NEF, VIF, VPU, and VPR. All accessory proteins are relatively small, ranging from 10 to 25 kilodaltons, and they are involved in regulation and viral pathogenesis. Post restriction factors, which restrict the effect of the HIV virus in the host cells, and if only when they inhibit the function of these restriction factors, they can cause infectivity in the host cells. So, which are the some of the important factors that restrict HIV infections have been discovered, including the PRIM, 5-alpha, ApoBec3, Tethrine, and SAN-HD1. Accessory proteins of HIV basically counteract with these restriction factors. We know that GAG, POL, and ENV are mostly responsible for the enzyme synthesis or different type of enzyme synthesis, whereas these accessory proteins are mostly responsible for counteracting the restriction factors to establish HIV infection. For example, like ApoBec3 is restricted or inhibited by VIF, tethrine by VPU, and SAMHD1 by VPX. So if we come to the structure of the retrovirus, basically it contains a RNA genome and an RNA directed DNA polymerase, which is the reverse transcriptase. RNA tumor viruses in this family mainly cause tumors of reticuloendothelial system, that is the liver, spleen, the small intestine. So these are the tumors of, they are called as a reticuloendothelial system and hematopoietic system. That is the leukemias and lymphomas or of connective tissues, that is the sarcomas. So here you can see that basically we can say here that basically the tumors which are caused are mostly of reticuloendothelial system and hematopoietic system, that is the leukemias, lymphomas or of connective tissues. Now coming to the properties of retroviruses, some of the virion, if you see here the structure of a retrovirus, you can see the proteins, then lipids are the present in the envelope part, and the enzyme, the reverse transcriptase, is inside the nuclear component of the retroviruses. In addition to that, there are certain viral enzymes which codes for the RNA or DNA, which codes for the genomic structure of the retroviruses. Now, virion is basically spherical, 80 to 110 nm in diameter, helical nucleoprotein within icosahedral capsid. Basically, two types of viral structure is there. One is the icosahedral and other is spherical structure. So here it is a spherical, 80 to 100 nm in diameter, helical nucleoprotein with capsid is the icosahedral. Now, composition is basically RNA constitutes about 1% of the total structure of the virus. Then protein about 60, approx 65%, lipid approx 30%, and carbohydrate to the 4%. Genome it is a single-stranded RNA. Linear structure of RNA is there, positive sense, 5 to 8 kilobase, and it is diploid in structure. So it is a single-stranded linear positive sense, 5 to 8 kilobase, diploid in structure. Molecular weight is 3 to 6 million Daltons. Protein present is the reverse transcriptase, which is present inside the virions, and envelope is present. Replication reverse transcriptase makes DNA copy from genomic RNA, and DNA provirus integrates into cellular chromosome. Provirus acts as template for viral RNA. So here from the provirus only the viral RNA is synthesized. So coming to the again to the HIV-1 virion structure. See, there are two types of HIV. One is HIV-1 and HIV-2. So we are discussing here about the HIV-1, which is the most common virus which is present, and HIV-2 is mostly in the African region. It is more common in the African part, but widely HIV-1 is present. So here you can see, and more or less they are same in structure, only that one of the accessory proteins is replaced in the HIV-2 in the case of HIV-2. In addition, it differs that in HIV-1 there is the accessory protein is the VPU, whereas in HIV-2 there it is VPX and genes that and RAV have two separate exons. Now here, so basically the difference is between the VPU and VPX gene between HIV-1 and HIV-2. So here, structural characteristics of a typical lentivirus retroviridae. It was originally named a retrovirus because of its particle-associated reverse transcriptase, a hallmark of the retroviridae family. It is a 100 to 120 micron enveloped virion with two single stranded. You can see here that there is two single stranded RNA, and here in addition to that there is associated with them is the it is a reverse transcriptase, and similar morphology to the lentivirus genus confirmed to its retrovirus. So here you can see that there are two single-stranded RNA genome to which the reverse transcriptase enzyme is associated, and covering them is an icosahedral capsid, and after the capsid region there is a matrix protein which is surrounding this capsular part, and then comes the envelope part, which is again made up of the protein structures and a lipid membrane. So here we can see, if we go to the detail of the structure, so HIV-1 virion uses the host cell membrane to form the viral envelope. Basically, the envelope structure is made when they infect the host cell, and this envelope part is derived from the host cell membrane. And the envelope is covered by two surface protein, that is the GP120, which is you can see here, which is outside the membrane, and another is the GP41. So this envelope is covered by GP41 and GP120 surface proteins, in as well as major histocompatibility class 2 proteins inserted into the lipid envelope or lipid membrane. Inside the lipid envelope, the matrix, which is formed by the GAG, we've already seen that two major proteins are Paul, GAG, and ENV. So here, this the matrix is formed by the GAG protein P17 holding the RNA containing core in place. The cylindrical core not only stores the viral RNA and various proteins, it also contains complementary RNA synthesized by the viral reverse transcriptase. Next, coming to the structure again, if we see in the detail, so in this you can see here that the outward structure is of GP120, and one stop is there of GP41, and inside there you can see the P17 matrix. P17 matrix is there, and then you can see here two single strands of RNA, and again the ENV GAG. Then again the three important enzymes are protease, integrase, and reverse transcriptase, which are present here inside the capsid part. Three important enzymes are protease, integrase, and reverse transcriptase. Again, you can see here that in addition to GP120, GP41, and P17 matrix, the capsid part is also called as the P24 capsid. So basically, two glycoproteins and two protein structures. So outward envelope is made up of GP120 and GP41. Matrix is constituted by P17, and capsid is constituted by P24. And three important enzymes are there, which are the protease, integrase, and reverse transcriptase, and two single stranded RNAs. Again, here you can also see that the lipid is a bilayer structure. In addition to that, other proteins which is present in the capsid is P6 and P7. So in detail, if we go through this viral structure, so it is more detailed structures. So in the next slide, in the next image, you can see here that there is a receptor. Basically, they infect the T helper cells present in our body. So here you can see that the virus attaches to the cell CD4 receptor present on the cell membrane, and the RNA genome is inserted into inside the cell by attaching to the CD4 receptors. Now we know that in the retrovirus there is a unique central dogma they follow, that first this RNA converts itself into the cDNA through the help of the enzyme reverse transcriptase, or other name is also called as the RNA dependent DNA polymerase, and they make a double-stranded DNA. Next, so the function of reverse transcriptase is to convert the viral host viral RNA, the viral RNA into cDNA, and the cDNA duplicates itself through the process of the replication. Now again comes the role of the enzyme, the integrase, and what integrase does, that it integrates viral DNA into the cell genome. So here the genomic is structure. So here you can see that this is the cell, host cell nucleus, and this double-stranded DNA gets integrated into the host cell genome with the help of the enzyme integrase. Now after the integration, see here, this black colored, more black colored, the viral DNA is integrated into the host cell genome. Then the transcription of the this transcription means the RNA DNA is converted into messenger RNA. Is the process is done, and then after when the transcription is done, then mRNA of the retrovirus DNA leaves the nucleus. After the process of transcription, leaves the nucleus and it enters into the cytoplasm, where again the process of translation takes place, that the mRNA is converted into the viral proteins. And here the role of protease enzyme, the role of protease enzymes comes, where it cuts up the protein, or you can say that it processes the viral protein, and again this viral RNA is reconstituted into a whole viral structure, and a new virus is formed, and again it is released outside the cell. So this is the entire cell cycle of the human immunodeficiency virus. Next here, so already we have discussed that it is very necessary to study about the how the oncogenicity is caused by the retrovirus or any tumor virus. So we have to go through the genome or the genomic structure of the virus. So if I compare, that is, this is the integrated DNA provirus which we have already discussed earlier, that with the virus, the viral structure genome which is attached to the host cell genome. So you can see here, this is they are processed and they are made into the 2 points are attached at the 5 prime and 3 prime end. One is this is the LTR. It is the long terminal repeats, and again long terminal repeats are again it is again made up of 3 parts, that is the U3R, U3 unique, U3 are repetitive, and U5 part. You can see at the all both the ends, LTR is made up of U3R and U5. Again here it is in the viral genomic RNA, there is no U3 part. There is only R and U5 part in the 5 prime capped end, whereas in the 3 prime end, you can see the U3 and R part. So you can see the difference between the proviral DNA and the viral genomic RNA. In addition to that, you can see here different accessory genes that we have already discussed. And in there, the three major active protein active gene is the GAG gene, Pol gene, and ENV gene. And it is again GAG is divided into MA, CA, and NC region, whereas Pol region is PR, RT, and IN, which is basically the protease code for protein. We will see in the detail in next slide that what proteins they synthesize here. So you can see basically what is the difference between the integrated DNA provirus and viral genomic RNA. So you can easily contrast the difference. Next, coming to the next slide, here you can see this proviral DNA that this is the GAG gene basically codes for nucleocapsid core proteins U3, but first let us discuss about the LTR part of the 5 prime end. So here U3R and U5, this is the bindings, LTR is the binding sites for host transcriptional factors. So basically here it is the this is the binding site for the host transcriptional factors. Next is the GAG gene, which is the nucleocapsid core proteins. Then Pol, which secretes for the reverse transcriptase, protease integrase, and ribonuclease. Next is the here you can see these accessory genes or some of the accessory genes are there. That is the VIF, VPR, and VPU. Here in HIV2, this only VPU gene is replaced by the VPX part, and it promotes infectivity of VIF promotes infectivity of cell-free viruses, whereas VPR is a weak transcriptional activator. And here VPU, you can see that is required for efficient virion budding. And here in V is the viral coat proteins mediated CD4 binding and membrane fusion. And in addition to that, there is a regulator of structural gene expression. REV is the REV response element, and this is the 3 prime end part. So coming to the next part here, if you can, we can see that between the viral RNA genome and provirus DNA. So in the viral RNA genome, 5 prime terminal is capped, then 3 prime terminal is polyadenylated, then a short sequence of R at each end, unique sequence U5 located near 5 prime end, while U3 at 3 prime end. U3 contains U3 contains promoter and enhancer sequences, whereas in provirus DNA is flanked at each end by long terminal repeat. Each LTR contains U3R and U5 sequences. Coming to the next part, that U3 and U5 are specific non-coding unique sequences found within the long terminal repeats at both ends of the retroviral DNA. They flank a repeated region. Already we have seen in the slide to form a U3R and U5 structure with U3 containing viral promoter and enhancer elements, and U5 is involved in the integration. Then coming to the transcription, splicing, and proteins, the promoter elements in the 5 prime LTR, which is the which is basically coded by U3 and which is coded by U3 U3 part of the LTR. So the promoter elements in the 5 prime LTR, basically U3 in the 5 prime, control transcription by cellular DNA directed RNA polymerase. The first polyase site in the R element is ignored, presumably because of the secondary RNA structures. At the end of the viral mRNA, the presence of U3 sequences upstream of R prevents this secondary structure, resulting in the polyase site being recognized to form the complete viral RNA. So when the viral RNA is formed, and the pro-virus DNA is formed. So we have already discussed the what is the major difference here. You can see here that 5 prime terminal is capped here, whereas pro-virus DNA is flanked at each end by LTL. Here it is capped and polyadenylated, where it is both 5 prime and 3 prime end is U3 and repetitive and U5 regions, which contains the promoter and enhancer regions. Then coming to the short sequence repeat, as each LTR contains U3, R5, and U5 sequence in pro-virus. Then here a unique sequence U5 is located near 5 prime while U3 at 3 prime. And U3 contains promoter and enhancer sequences. So viral mRNA is then spliced, fully spliced mRNAs for the accessory proteins, that is the REV, TAT, and NEF. Now we will see what are the role of these accessory proteins in our next slides. Then cellular processes prevent unspliced RNA from leaving the nucleus. So whatever the cellular process is going on, it does not prevent the unspliced RNA leaving the nucleus. That is, after the transcription, we have seen that it leaves the nucleus into the cytoplasm where the process of translation takes place. But here, if this splicing basically that it converts into from the RNA to mRNA by exons and removing the introns part and exons part coming together, which codes for certain triplet codons. But viral REV allows export of incompletely or unspliced mRNA from the nucleus. Incompletely spliced mRNAs encode ENV, which is cleaved into the envelope proteins SU and TM and the accessory proteins VIF, VPU, and VPR. Unspliced full-length mRNA serves as genomic RNA that is packaged into virions and or as a template for translation of the GAG and GAG whole polyproteins. Ribosomal, it is frame shaped. So here, if you see in detail here, you can see here that basically the GAG gene is the proteins encoded is of P24 and P7 and P17, and they are basically forming the nucleocapsid and in the formation of matrix. Pol is the reverse transcriptase, protease, integrase, which reverse transcriptase, as we already know, that it transcribes RNA genome into DNA. Protease cleaves precursor polypeptides, and integrase integrates viral DNA into host cell DNA. ENV codes for GP120 and GP41, which is attachment to CD4 protein fusion with the host cell. It is basically GP41 is responsible of the fusion with the host cells, and GP120 leads to the attachment to the CD4 protein. Second is the regulatory genes which are found in HIV that are required for replication is the TAT gene, which codes for TAT protein, which is activation or transcription of viral genes, whereas REV gene codes for REV, which transport to late mRNAs from nucleus to the cytoplasm. So this is the regulatory genes which are formed. Then we can see here that GAG, again, we have seen the three components, that is the MA, C, and NC, basically matrix, capsid, P24, and nucleocapsid proteins are synthesized by the GAG. Pol, PR, RT, iron, we have already discussed about it. Then protease, it is aspartate protease, reverse transcriptase, integrase. So these are the functions. ENV, surface proteins. ENV codes for the envelope part, which is constituted by SU and TM. So here surface protein is the GP120 and transmembrane protein is the GP41. Surface protein helps in receptor binding, whereas transmembrane protein, that is GP41, helps in the fusion domain. So here we will see the accessory proteins. Till now we have discussed about the regulatory proteins and the proteins. Now what are the accessory proteins which are present here? The VPR, which is the viral protein R, and it causes the G2 arrest. G phase, S phase, there are different phases in the cell cycle. So it helps in the G arrest in the G2 phase of the cell. TAT, that is the transactivator of transcriptase, which is a transactivator, REV regulator or virion protein, which is the RNA export. VIF, virus infectivity factor, which is ApoBic, 3G inhibition. We have seen that some of the inhibitory effects by which is given inhibitory effects shown by the cell host cells, in which one of the was the apobec 3G, which does not allow the viral genome to transcription transcribe. So here it is basically accessory proteins which negate the effect of these inhibitory factors of the host cell genome. So these are some of the important proteins. Now here we can see that the NEF gene, which is protein NEF coded NEF, so it decreases CD4 proteins on class 1 MHC3 and MHC proteins on surface of infected cells. So as these are decreased, so what it induces? It induces the death of uninfected cytotoxic T cells. So it does not allow the cell to respond against the foreign invasion by decreasing the CD4 proteins and class 1 MHC proteins, and hence it goes unrecognized and escape the very mechanism of immunity. Next is the VIF, or proteins encoded is VIF. It enhances infectivity when inhibiting the action of apobec 3G, already discussed, enzyme that causes hypermutation in retroviral DNA. So it inhibits the apobec 3G is mostly responsive, mostly helps in, does not allow the retroviral DNA to transcribe and by through the hypermutation. So it is the VIF gene which is present in the retrovirus. It does not allow apobec 3G to function by making it non-functional by inhibiting it. Then comes the VPR gene, which transports viral core from cytoplasm into nucleus in non-dividing cells, and VPU, which enhances the virion release from the cell. Unless and until it is not released, it will not be able to infect the other cells. So it is supported by the VPU protein synthesized by the gene VPU. So in addition to the functional genes, these accessory proteins are holds a very, or we can say regulatory genes, which holds a very important role in the multiplication and increasing the infectivity and release of the cell from the host. So this is the role of some of the accessory proteins which are present. So we can say that retroviruses are RNA virus that reverse transcribe their RNA into proviral DNA, and these viruses are associated with a wide variety of cancers in chickens, mice, cats, and monkeys. Some retrovirus cause specific type of cancer soon after infection in high proportion of animals, whereas others cause various cancers late after infection in a lower proportion of animals. Highly oncogenic retroviruses are recombinants of viral and host genes. See, what it does, it means that usually new viruses which are released, they, as we have seen already discussed, that our cell or host cell also contains some proto-oncogene, or which is converted into oncogene, or the cellular gene also contains some anti-oncogene, which in which anti some anti-oncogene which negates the effect of the gene which suppresses the activity of these oncogenes. So while during the while the when the this multiplication, as we know, they integrate the proviral through the help of proviral DNA. If you have studied about the lysogenic cycle or lytic cycle in the genetic part of the bacteria transformation, so you have seen that in the lysogenic cycle, the DNA of the bacteriophage or any cell gets integrated into the host cells, and when they start multiplying, so they carry some of the host cell genome also. Similarly, here also it is discussed that and they become recombinant. So here it is the new viral genome which comes out of the host cells is a recombination of both retroviruses and host cells. It is not the purely that only the retrovirus genome will come, but it will also include some part of the host cells. And the cancers induced by these viruses are determined by the transduced host gene. So retroviruses that cause cancer at a low incidence do not contain inserted host information. Rather, they appear to cause cancer via mutation of the expression of potentially oncogenic host genes. So it is basically due to they do not cause cancer, but due to as they are carrying some host oncogene in between among themselves. So any mutation of the expression of potentially oncogenic host genes leads to the causes the oncogenicity or induces a property of oncogenicity in this DNA in these viruses. And during the normal life cycle, retrovirus integrate proviral DNA into the chromosomal DNA of their host. Integration occurs at many different sites. Most integrations are benign, means that it is not so not effective or not cancer causing, but some results in cancer. Retrovirus research has identified 12 potentially oncogenic host genes. So that's why I was telling you that it is very necessary to learn about the genomic structure of any retrovirus to understand, or any tumor causing viruses, because only through the study of genomic structure we can understand how they have converted themselves, or how this cause oncogenicity or cancer in the host cells. So here it is said that when the retrovirus research has come to the conclusion that there are basically 12 potentially oncogenic host genes which are present in the retrovirus, and each is associated with a characteristics cancer or a spectrum of cancer. So this is the main reason of concern, that these 12 potentially oncogenic host genes can cause 12 different type of cancers in the host, and several appear to code for protein kinase that phosphorylate tyrosine residues. So we can see here that what is the replication of retroviruses. So here the viral RNA serves as the template for the synthesis of viral DNA through the action of viral enzyme functioning as an RNA-dependent DNA polymerase. By complex process, sequences from both ends of the RNA become duplicated, forming the long terminal repeat located at each end of the viral terminal. Long terminal repeats are present in viral DNA. Only in viral DNA, the newly formed viral DNA becomes integrated into the host cell DNA as a provirus. Progeny viral genomes may be transcribed from the provirus DNA into viral RNA, and the proviral RNA is transcribed to the host enzyme by the host enzyme by RNA polymerase 2. So basically, here we can say that the transcription process is of the viral DNA. Mostly, this viral DNA utilizes the host cell enzyme itself, that is the RNA polymerase, to convert itself into the from proviral DNA to viral mRNA. And here one important point to note is that the virus happens to contain a transforming gene, the oncogene plays no role in replication. So here there is no oncogene, does not have any role in replication, and this is marked contrast to DNA tumor viruses, where in which the transforming genes are also essential for viral replication genes. So this is the two opposite characteristics in the DNA tumor virus. Oncogene is necessary for its replication, where no such role is played in the RNA tumor virus. So if oncogene is present or not, it does not, it will not affect the replication of the virus. Coming to the oncogenicity, the properties, so retrovirus that contains oncogenes are highly oncogenic. They are sometimes referred to as acute transforming agents because they induce tumors in vivo after very short latent periods and rapidly induce morphologic transformation of cells in vitro. Briefly, neoplastic transformation by retroviruses is the result of a cellular gene that is normally expressed at low, carefully regulated levels becoming activated and expressed constitutively In acute transforming viruses, a cellular gene has been inserted by recombination into the viral genome and is expressed as a viral gene under the control of viral promoter. So here basically in the recombination, as we have discussed here, so the cellular gene is basically inserted by recombination into the viral genome and is expressed as a viral gene which is under the control of the viral promoter. So in the case of slow transforming leukemia viruses, the viral promoter or enhancer element, which we have seen in the LTR part, that U3R and U5. So it is basically this oncocellular gene is expressed as a viral gene and it's under the control of this viral promoter, and enhancer element is inserted adjacent to or near the cellular gene in the cellular chromosomes. So these are some of the important points which are related to the oncogenicity of the retroviruses. And with this, and we can see here that how important is the study, so that we can come to the conclusion that how the oncogenicity, though it is a only two viruses, that is the retrovirus and the hepatitis C virus, flaviviridae, belonging to the family Flaviviridae, are the two important RNA viruses which causes oncogenicity, but their the effect on the or their consequences on the tumor formation is very large. So with this, we conclude our today's lecture. If you like my lecture, please like it on my channel. Thank you.